I'm Tim Ventura, and we're joined today by Dr. Stephen Benner, known for his work in synthetic biology, including creating in the lab DNA that might be found in alien life forms, Jurassic Park experiments that resurrect proteins from ancient Earth life, and generating COVID test kits that were used in India, Europe, and the US, among many other contributions to science. Dr. Benner has been a professor at Harvard University, ETH Zurich, and the University of Florida, where he was the VT and Louise Jackson Distinguished Professor of Chemistry. In 2005, he founded the Westheimer Institute of Science and Technology, the Foundation for Applied Molecular Evolution. Dr. Benner and his colleagues were the first to synthesize a gene, beginning the field of synthetic biology. He was instrumental in establishing the field of paleogenetics, interested in the origin of life and the chemical conditions and processes needed to produce RNA. He's worked with NASA to develop detectors for alien genetic materials using the definition of life developed by the NASA Exobiology Discipline Working Group in 1992. He has a BA, an MS in molecular biophysics and biochemistry from Yale, and a PhD in chemistry from Harvard, which has been awarded over a dozen awards and honors related to his research. So, Stephen, welcome back. Welcome back. That is an enormous introduction, but you have so many accolades and awards that I just felt like I couldn't skip any of that. So,
oh, thank you very much. I've just heard my obituary, so we can move from there.
well, so much of that experience is relevant today, though. So in our last interview, we were talking about synthetic DNA, and you actually surprised me when you started talking about life on Mars. And I mean, I just got so incredibly excited about that. So it is truly an honor to have you speak on this.
Well, thank you. I mean, of course, it's true that if you have life on Mars, it need not have the DNA that we have here on Earth, you and I. And so it's quite possible that what we synthesize in the laboratory as alternative forms of DNA could very well be found naturally in an organism on Mars that does not share our natural history.
The video that I referenced was a presentation that you gave called "The Case for Extant Life on Mars" at the 25th Annual International Mars Society Convention. So this was last November, and I will put a link into your presentation video for the audience. And again, highly relevant. I mean, robotic exploration of Mars has been going on for decades. But really, in the last decade, innovators like Elon Musk have really started pushing the colonization narrative back into global headlines. And I think that just makes this idea and this question of extant life incredibly relevant today. So I want to start by asking, what was your inspiration for last November's presentation?
Well, I was asked to give a talk on the case for extant life on Mars. There's a separate line of research that we do here, which asks how life could have emerged on Earth. And there is a model for, there are many models for how life might have emerged on Earth, but one of the most coherent models says that in a geological environment, roughly 4.2 or 3 billion years ago, chemical processes occurred that led to the formation, a biological formation of RNA, ribonucleic acid. Now, you know about RNA these days because it is the genetic material of the coronavirus that caused COVID, which of course is a unfortunate use of the RNA molecule, at least from our perspective. The virus has a different perspective, I suppose. But yeah, so the thought was that if you can get RNA to emerge from a geological environment, a prebiotic soup, you would then have the informational molecule that would start Darwinian evolution. And from there on out, you have Darwinian evolution at your disposal. And so almost every problem in creating life-like behavior becomes much easier. So the real major problem is that very first step to get you out of normal chemistry, which normally causes organic molecules to degrade into tars and asphalt, into something that has access to Darwinian evolution. And that is a process involving the reaction of common molecules that must have been present on what we call the Hadean period. This is the period of time before life emerged, at least as far as we can tell, to get you RNA. So it turns out that from that line of research, there's a certain class of rocks, lava, basalt rocks. You might, if you go to Iceland, you will see them coming out today. If you go to Hawaii, the same. But of course, you can, if you live in New Haven, you can look at East Rock and West Rock. Those are lava basalts. They were certainly present in the Hadean. And what we had shown or discovered was that those rocks would allow the formation of RNA if you had the correct precursors. And those correct precursors could come also from this kind of basalt lava. If you had certain other kinds of chemistry that was going on in an atmosphere that had just suffered an impact of a reasonably sized asteroid body.
Okay.
So that creates in the atmosphere a reducing atmosphere because the asteroid's coming in has a lot of metallic iron on it. So it reduces all the oxygen and carbon dioxide to methane and hydrogen cyanide and very useful molecules to create the bases, the nucleobases, the A, U, G, and C, the 4 letters that we use in our RNA to encode information. And that reducing atmosphere is what we like to say in chemistry, out of redox equilibrium with the stuff going on the surface. That is, the stuff on the surface is more oxidized, the stuff in the atmosphere is less oxidized. And furthermore, the surface has access to borate minerals. This is like borax, which you use in your laundry to wash clothes. 20 Mule Team, for those of us who are old enough to remember Ronald Reagan. Of course, he got his career started on television by advertising borax from Death Valley, actually. But these we were able to show that these go all the way from carbon dioxide in the atmosphere, all the way to RNA on the lava rock. And that's great, but then it turns out that Mars also has lava rock. In fact, almost all of the surface of Mars that's accessible to bedrock is basalt lava. We have elemental analyses of that rock, which says it has boron and phosphorus in the right oxidation state and all the rest. And so all of a sudden, what we have shown in the laboratory on Earth as a way of getting RNA as an informational molecule out of prebiotic stuff on prebiotic rocks, all could have happened on Mars as well. So at that point, you go to the Mars Society, and the Mars Society is it's a dues-paying society where people, pay dues to go once a year to meet other people with like-minded views of Mars and an interest in Mars. And you say, hey, all the chemistry that we think is important for the origin of life on Earth could also have taken place on Mars. And so life is likely to have emerged on Mars, or it's likely also to have emerged on Earth or Mars and then been kicked out by an impact, traveled on a rock to the other planet and seeded life there. So it's possible that Earth life and Mars life share a common ancestor.
Well, and that actually takes me deeper into the questions. But when you mentioned borate, that was something in the presentation you talked about that, right? So RNA itself is not entirely stable, I guess, in the natural environment, and it helps to have chemistry to stabilize that. And you were saying that borate in the rocks is more prevalent in Mars. And so Mars might actually be more hospitable to the creation of this than it was here on Earth.
Yeah, I mean, there's a little subtlety to that comment. It's the precursors for RNA that are unstable. And these precursors are carbohydrates. They are sugars in RNA. The sugar is ribose. That is a sugar with 5 carbon atoms in it. You're much more familiar with sugars like glucose, which have 6 carbon atoms in them. But the point is that normally sugars, if you heat them, turn into caramel, they turn into goo and sticky stuff, and that's going away from life. But when you have boron minerals like borax in the environment, you prevent that. And so the precursors for RNA become stable in the presence of borate minerals. And more to the point, borate minerals bind to various intermediates in the processing or the natural reactivity of those sugars and direct them in a way to form ribose and then ribonucleotides, as we like to say, and from there to RNA. So yeah, absolutely. And then the other problem is that boron is not a popular element. If you go to your periodic table book, you'll find that boron is actually quite low in abundance. And the reason for that actually is the same as why they use boron in control rods and nuclear reactors. boron absorbs neutrons and becomes something else. Actually, it becomes carbon. So your problem is getting enough boron out of the sun when the sun's got nothing but neutrons flying around. So very little boron is existing. So if you just had the average amount of boron, you wouldn't have enough. But then boron doesn't go to the core of the earth like iron does. Boron remains on the surface. And now that's fine if the earth is flooded with ocean, boron gets diluted into a global ocean, but Mars was never flooded. So you always have the possibility of getting more boron on the surface of Mars than you have on Earth. And in fact, there's a guy named Patrick Gazda who, did some analysis interpreting data that was collected by rovers that NASA sends on Mars, and they found a lot of boron. So as far as we're concerned, the amount of boron that is necessary to form life was there on Mars, not diluted into a global ocean. And so in many ways, Mars is a more hospitable place to start life than the Earth was.
On that note, let me back up to the Viking probes in 1976.
Yeah.
And again, you'd mentioned this, I think, at the end of our last interview, and this is what got me really excited. So you mentioned an experiment in the Viking mission back in 76 that appeared to show microbial activity in the Martian soil. If I understand this correctly, and I went through the video twice trying to write everything down from your presentation, it looks like they performed actually 3 tests as part of this experiment. One was looking for microbial metabolism, another was looking for photosynthesis, and then there was a third looking for metabolic gas exchange. And all 3 of these experiments produced positive results, right?
That's right. And Gil Levin, who designed the experiment where he put food onto the surface of Mars, where the carbon in the food was radioactive, and then saw the evolution of radioactive carbon dioxide. By the way, that's what would happen if I gave you radioactive food. You would exhale radioactive carbon dioxide. So they saw that result. He died about 2 years ago at the age of 97, but at the end, he still was convinced that he had discovered the first evidence for microbial life on Mars by discovering a oxidative metabolism that is analogous to what you would do with the same kind of food.
So that's 3 positive results. And then if I understand correctly, the question that they had was, and again, this was in your presentation, these metabolic results could be from microbes. They could have potentially been from natural processes. So NASA it almost sounded like kind of a tiebreaker experiment, I guess, looking for reduced carbon with the GS mass spectroscopy, I guess. And that yielded no results, even though reduced carbon should be naturally deposited on the surface daily, right? So I think, yeah, it gets a little complicated there. And I had difficulty describing that, but I think the idea was, it didn't show carbon, so they said the surface chemistry must be so reducing that carbon that reaches the surface is immediately, so oxidizing that carbon is immediately chemically changed, I guess. Would that be correct?
Yeah, well, that's right. So the concept of a tiebreaker is a reasonable one. So it's an amusing story. If I give you if I give you food that's radioactively labeled carbon, you will exhale radioactive carbon dioxide because you oxidize the food. You eat the food. Now, if I give an automobile radioactively labeled gasoline, it will also emit carbon dioxide. So your core question is going to be, we observe the release of radioactive carbon dioxide. Is it coming from a living system, Tim Ventura, or is it coming from a non-living system, an automobile? Now, keep in mind that the automobile is a biosignature. It required a human to build it, but for the time being, we can use that as an analogy for some non-biological process that will also burn food. And so that's always going to be the problem. You can try to find alternative non-life explanations for the gas exchange data. They saw a release of oxygen from the surface. You can see the same thing for what was technically a photosynthesis type of experiment. If I go outside with my plants and I put radioactive carbon dioxide into the air, this is the other direction, the plant will make sugar out of it. It'll make, that's what the plants do. And so the argument was that they actually observed that also on the surface of Mars. But in each of these particular cases, the experiment that could be interpreted as a sign of microbial life on Mars could also be interpreted as a sign of non-microbial, non-biological processes that produce the same result. So then, now there was a gas chromatography GC mass spec, and what that's a big name for just a very simple process. They scoop some soil into a little cup. They heated the cup up to 400, 500 degrees centigrade, hoping to see organic material break down into a gas form. They would then separate the gases, and they would try to detect in that gas mixture organic molecules by mass spectrometry is the technique, but what they're looking for is ions that come from organic molecules. Now, it's not quite true to say that they found nothing. They found compounds that they did not understand, one of which was chlorinated hydrocarbons. This is compounds with carbon and chlorine in them. And that was not an expected result at all, because if I took Tim and put you into a cup and heated you to 500 degrees, I would certainly get organic gases out of you and detect them, but I would not have much that had chlorine attached to the carbon.
Okay.
Now, the poor people in 1976 did not know what to make of this. They actually did reproduce the experiment at 2 sites, because Viking actually was done in duplicate. They landed the same package in 2 places in case one of them failed. And they also had tested the machine on the flight out. So they had seen some of these compounds, not all of these compounds, on the way out. That was good, because that told them that the machine was actually working. But then the other problem is back to what you just said a moment ago. We know that organic molecules fall to Mars all the time as meteorites. They come to Earth all the time. You can go collect them. You can buy them on eBay. These are called carbonaceous chondrites because they contain carbon. And the question, in fact, the machine before it flew was criticized by various people who said, wait a minute, all you're going to do is detect organic molecules that have fallen to Mars from outer space. And that's not interesting, and you're going to be deceived by them. And so when they saw very little of this meteorite organic, in fact, very little of this carbon at all, and all they saw was compounds containing chlorine, they said, oh my goodness, this Martian surface must be self-sterilizing. It must be oxidizing the meteoritic compounds that fall. And of course, if it's oxidizing the meteoritic compounds, if I put Tim onto the surface as well, he'd get oxidized also. So this argued that the soil was inhospitable to all life because all life requires organic molecules. We didn't find them. Now, my only role in this was in 1999. I went through the mission logs and I said, wait a minute, they didn't look correctly for the organics. They did not look with a tool that was sensitive enough to detect them. And so I published a paper on this. A couple of years later, a NASA mission found a salt on the surface of Mars, not sodium chloride, which is the salt that you and I are familiar with on our tables, but sodium perchlorate, which is a chlorine atom plus 4 oxygen atoms. And it turns out, just as with fireworks, by the way, perchlorate is what they make fireworks out of, because if you heat perchlorate, well, if you put perchlorate at room temperature, it does nothing. In fact, you can drink a solution of, I don't recommend it, but it's not going to oxidize you. But when you heat it to 500 degrees, you get a firework. You burn all the organic molecules, and what you don't burn, you make into organic molecules that are attached to a chlorine atom. So all of a sudden now we know entirely what was going on with the Viking gas chromatograph mass spectrometer, is that they were scooping organics into a cup with perchlorate in the soil as well. They were heating up to 500 degrees. They were doing some pyrotechnics in the cup, and then they were looking at the products of having toasted fireworks. Okay, so basically you will get some carbon out of fireworks, but you won't get anywhere near the amount of carbon that you had when you started if you were using fireworks that contain carbon. And so, and then what's happened since then is that rovers have trotted it around Mars and Jezero crater and these other places, and they have found organic molecules. And we had proposed that one of the molecules that they should find would be benzoic acid.
Yeah.
It's actually found in your food stuff as a preservative in some foods that you buy at your local grocery store. It happens to be somewhat oxidized, but stable to further oxidation. And one of the things that they found about 3 years ago was benzoic acid. So at this point, the chemistry has won. We no longer can rule out biological explanations for the Viking results by saying that the surface is so oxidizing that it is self-sterilizing and cannot hold any organics at all. We now know that the Viking GC mass spec data were misinterpreted. We know why they were misinterpreted. We have an entirely consistent result for their results, which, an entirely consistent explanation for their results, which involves this combination of organics, which were there, and perchlorate, which was there. And we have now gone back and actually shown that there are organics there, so the surface is not self-sterilizing. So now you're back to, now poor Gil Levin kept saying, well, wait a minute, my experimental interpretation was ruled out by a thought that the surface of Mars was self-sterilizing and oxidizing, like bleach. Benner in 1999 showed that that was not the case based on the mass spec result analysis. Chris McKay at NASA Ames did some lovely work a decade later, which really sealed this story up as I've just described it. So Gil says, well, wait a minute, why don't we go back to the status quo ante and say, wait, Gil Levin discovered life on Mars through his metabolism experiments.
Well, and as you mentioned, so benzoic acid, and then one of the other things he mentioned was, so NASA's Curiosity rover found evidence of organics in 2013. Perseverance found organics in both Jezero crater and Wildcat Ridge. And again, 2021, I guess, was when the benzoic acids were found in the Martian dunes. So we have positive results from Viking initially, further results from the rovers, and then the only negative result was the GS mass spec, which has been, I guess, ruled out because of it just not being sensitive enough.
The GC mass spec, actually, not to correct you, but yes, exactly. And of course, the problem is they found benzoic acid in the dunes. Benner says, oh, this is great. I predicted that. But hang on. Okay, that now could be coming from meteorites. It need not be coming from life. So here you are constantly, you're walking through the world in a fog. Every result that you can interpret this way can also be interpreted that way, including the fact that we are now finding organics on Mars. But the logic, if you just are Aristotle here and don't know any about the substance, it's just writing down what you hope to be a syllogism, the logic is that you can no longer rule out Gill's life interpretation, life on Mars from the 1976 results, by saying that there are no organics on Mars. There are organics on Mars. Okay, so we're back where it's possible that Gil Levin's 1976 Viking experiment discovered life. So you're right back to where you started. And now the problem will be to try to design definitive experiments. The problem has been with NASA is that the NASA has been very hesitant since Viking and the Allen Hills meteorite and other experiments on Mars to ever launch a mission for explicitly designed to detect extant life. So you would talk to NASA in 2000 to 2020, the program people are at NASA headquarters are perfectly honest about this. They say they're afraid that if you announce that you're going to look for life, that's what we did in 1976. The results were ambiguous, and the program within weeks was shut down, and we go into a 20-year hiatus where no Mars explorations at all is being done.
Okay.
So this is a sociological problem. How do you keep a nation committed to looking for life? Well, you don't say you're looking for life, so you can't be disappointed if you don't find it or don't find it unambiguously. You say, I'm going to look for water. So NASA said, follow the water, or that's what these missions are designed to do. They're going to places where there was past water. You go look for habitability. That is an environment which might hold life. Okay. But you don't anymore tell the public, I'm going to go search for life. You go fly drones on Mars, right? That's great fun, right? Everybody can relate to that. But so a lot of us in the community who are quite convinced now, as I've just described to you, or described to the Mars Society, that life could have emerged on Mars. There is a problem. Mars has changed. So has Earth since then. Did that change drive life on Mars to extinction? Or are you like Jeff Goldblum in Jurassic Park saying, life finds a way, right? So on Earth also changed. You have all this oxygen in the atmosphere, which is poison, right, to any organism that hasn't figured out how to handle it. So the question is whether Mars life found a way to remain to the modern day. And so we're very annoyed that they're flying drones on Mars when they should be looking for life on Mars, because we have such tantalizing evidence that is consistent with a possibility of a Mars biosphere, and NASA doesn't go looking for it. Very, very annoying.
And you brought up another point, which was that Mars potentially once had a thick atmosphere, and I've read standing bodies of water. They didn't say if that could be like lakes or oceans, but at least enough water to support life and support the evolution of life. And so that changed over time. My understanding is just that it doesn't have enough gravity to retain that atmosphere. So the atmosphere just boiled off over time. But so Mars.
But yes, Mars undoubtedly had a thicker atmosphere initially. It undoubtedly had bodies of water. You can see lakefronts, you can see salts that were precipitated. The Mars, as you mentioned, has a gravity that is much less, well, not much less, but significantly less than Earth, so it doesn't retain the atmosphere quite so well. And also, the atmosphere is carbon dioxide. And so what happens is that the rocks, the basalts, are alkaline, and they will absorb carbon dioxide from the atmosphere. So the 3% nitrogen's there, carbon dioxide is there, but it's constantly being sucked out by the alkaline rock. So so Mars lost most of its atmosphere. So right now, the average atmosphere of Martian atmosphere pressure is roughly what we call the triple point of water. This relates to the fact that when you lower the pressure, the boiling point of water goes down. So if you go to Denver, 1 mile high, famously, right, the boiling point of water is lower than 212 Fahrenheit, less than 100 centigrade. On Mars, you're down at a pressure which almost is exactly the pressure at which water cannot exist as a liquid, only as ice or as a gas. But the Hellas Basin is lower in altitude. So it's like the Dead Sea. So we can have liquid water there, but just intermittently. So yes, you've gotten to the point where the pressure of Mars is almost exactly balanced between the pressure at low altitudes where water is liquid, but it's possible to have liquid water, and at high altitudes where it's not possible to have liquid water. And that's where Mars is right now.
Well, one of the things that had occurred to me was finding life on Mars raises a lot of questions about quarantine also, right? I mean, the first thing that I would envision happening is, obviously there would be a media circus, which NASA doesn't want. And I would assume that environmental groups would push for as strict quarantine as possible to prevent damage to the Martian biosphere and contamination. But then the reverse is also true, right? We have programs like the Mars Sample Return Program and things like that, where if Mars is a dead planet, it's safe. But if we view it as having its own life, I guess that could provide some dangers, bring some of that back to Earth, right?
Yeah. So you're worried about reverse contamination. That is, if Mars has a biosphere, right? It is conceivable that when we bring a sample of Mars back from Mars, it will bring with it bacteria that will then contaminate the Earth. But here is a sample of what's a meteorite called Black Beauty. I'm now holding it in my hand. It's 5.5 grams of Mars. It has traveled from Mars to Earth, where it landed in the Sahara Desert, where it was picked up by some camel herders who are fully aware of the fact that if there's a rock on the surface of a sand dune, it fell there. And they know that there are all sorts of Westerners, myself included, who will pay him good money for that piece of rock. That rock was never heated. So the rock comes in by meteorite, and it's true that the outside of the meteorite is heated, but that material gets blown back, ablated. And so this rock lands without ever having been heated above 40 or 50 degrees centigrade. So if there's any microbes on this rock, they have survived. And this, by the way, goes as a traffic that occurs because something has hit Mars. We actually have a reasonably good idea where this came from on Mars. Some meteorite hit Mars. It was big. It then ejected rocks. Some of the rocks escaped the gravity of Mars and traveled around the solar system for a while before they landed in the Sahara Desert. There's the Allen Hills meteorite in 1996, which had ostensibly cell structures of Martian life, which landed in Antarctica. Again, never been heated. It was not heated upon ejection. The inside was not heated when it landed in Antarctica. It has been kept quite cold ever since. And so there's this intercourse going on back and forth between Mars all the time. So the fact that I have actually 5 grams of Mars in my left hand, it's a sizable fraction of what NASA will bring back from Mars. But of course, NASA will bring this back to a top security station on the moon, with worried about quarantine and all the rest of it. Our view is that if there was any possibility of Martian life coming to Earth and killing us all, it already would have done so. Martian life has been, to the extent to which it exists, has been coming to Earth all the time, maybe 500 kilograms of this stuff comes to Earth every day. So the concern about backwards contamination from Mars to Earth, in my view, is a little bit misplaced because it's already happened. Now, going in the other direction, it's harder to eject rocks out of Earth's gravitational field because Earth has more gravity, right? And also Earth has more atmosphere, so it's harder to get stuff. But when that meteorite hit in 65 million years ago and extinguished the dinosaurs, some of the stuff was ejected from Earth's gravity. Some of it landed on Mars. So there you could probably find a little bit of dinosaur bone in the Martian surface if you knew where to look, and we're extremely lucky to find it. But yeah, you can go across the surface of Mars and you can look at the surface of Mars and see all kinds of meteorites sitting out there. Some of them are from Earth.
Yeah. Well, and one of the exciting things about this is it brings up this idea of panspermia, right? Where life could have originated here, could have originated on Mars, or some people have postulated that maybe it originated inside of comets, right? Which are mostly water as they get close to the sun. You've got all these chemical reactions in liquid water, and the rest of the time it's frozen. So
yeah. So back and forth between the planets in a star system is reasonably common. Now, of course, you don't get anything out of Jupiter. Jupiter just is too big. So, and it's also too liquid, right? Jupiter is basically a gas giant. And of course, interstellar panspermia, like what Ari is talking about at Harvard, is of course a very, very much lower probability event compared to Mars and Earth. But the fact that I can show you in my left hand 5 grams of Mars, which we had a donor named Bob Bruner in Denver helped us buy, was very nice of him to help us acquire this. But this is a couple $1,000s worth of rock, and so there you are. So the fact that I can hold it means that we have already done sample return from Mars.
Well, and again, it just in terms of big picture ideas, I think it's amazing to pause and think about basically the planets sharing life, sharing rocks. We tend to think of these as discrete entities, right? Where they're completely separated from each other. But, and in the short term, that's true. But in the longer term, things do travel from one planet to the next.
Yeah. There are pieces of Earth on Jupiter.
Yeah, amazing. Well, and Jupiter's moons as well. And
Absolutely.
they postulated that there could be liquid oceans underneath those. So
absolutely. So Europa has a liquid ocean. There are pieces of Earth that landed on Europa.
Yeah, absolutely amazing. Well, Stephen, let me thank you so much for your time today. Again, it has truly been an honor having you with me. And I appreciate your explaining this for the audience. I hope I did well setting up the questions. This was definitely a little bit more complex than normal, but it's an absolutely wonderful topic. And I think it goes to so many of these ideas about life, right? I mean,
It does.
where it originated and how it evolved, and I mean, how it may have gained a foothold on all these different worlds. Uh,
Absolutely.
so if it's okay, let me close by asking what you see happening in the next few months with this story, because your presentation was last November. And so it sounds like all of this information is starting to come out very recently, right? It's all coming out rapidly and recently. There were recently soils that were demonstrated to be able to grow Martian crops. So there's a lot happening here. Do you expect any developments in the near future?
Well, it's an interesting question. The obviously NASA operates on a 10-year time scale. There is a group of people who's been gotten together by a colleague of mine named Jan Spacek, who has started something called Alpha, the Agnostic Life Finding Association. So you can go and it's a again a volunteer society like the Mars Society, but alphamars.org, I think, is their webpage, is a place you can go and join a community of people who are really now desperately trying to encourage NASA to actually go look for life as opposed to just flying drones or looking for water or looking for habitability. So that's there. So, and then as you've already mentioned, SpaceX is a private organization which has a publicly stated commitment to go to Mars as well. And so it's quite conceivable that citizen groups like Alpha Mars and SpaceX will at some point get together and figure out how to do a joint mission where they go to Mars. I think SpaceX wants to put people on Mars sooner or later, but they're going to have to go before then and robotically mine water, for example. That's water will be used before the astronauts arrive to create propellant to fuel their trip back to Earth, and that will be done robotically or before astronauts arrive. But I know Alpha Mars and Jan has a whole scheme out there saying how they would look for a biosphere in the water that SpaceX will be mining to prepare for human visitation. And the Alpha Agnostic Life Finder design is in principle an instrument that has value as a low cost add-on to a water mining operation on Mars, where the tons of water are a biological sample of Mars. And water is being mined on Mars in ton quantities because you need tons of propellant to get back to Earth. But to set astride that water mining operation and agnostically look for life in that sample. And agnostic in this context means looking for life in that sample where we do not assume, as we do the looking, that the life has the same natural history as Earth life and may not even have the same core molecular biology as Earth life. That would be the kind of thing that SpaceX and the citizen groups could get together and do quite productively. So that's what I'm expecting to see in the next year or so.
Wonderful. Thank you again so much for your
My
time
pleasure.
today, sir.
My pleasure.